What are the molecular secrets of tardigrade survival? Imagine a tiny creature that can survive being blasted by radiation, frozen solid, or dried out to a crisp. That's what makes tardigrades so fascinating in the world of bizarre biology. These water bears are tiny, just a millimeter long, but their survival tricks are anything but small. So, what are their molecular secrets that let them pull off such incredible feats? Well, it all comes down to some special proteins and DNA protection methods they've developed over millions of years. One of their most unique tools is a protein called Damage Suppressor Protein, or Dsup. This protein acts like a shield for their DNA, attaching directly to it and preventing damage from harmful radiation and other stresses. Scientists have even inserted Dsup into human cells, and those cells showed much less DNA damage when exposed to X-ray radiation. That's a big deal because it shows how powerful this tiny protein really is. But protecting DNA isn't enough. Tardigrades also have a remarkable ability to repair their DNA after it gets damaged. They produce a protein called TDR when that binds to broken DNA fragments and helps organize them into compact structures. This process makes it easier for the tardigrade cells to fix the damage quickly and efficiently. Unlike most animals that relying on damage-specific repair proteins, tardigrades physically manage their DNA fragments to keep their genome stable. Another key part of their survival toolkit is a group of proteins called Intrinsically Disordered Proteins, or IDPs. These proteins don't have a fixed shape like most proteins. Instead, they change shape depending on the environment. Tardigrades produce IDPs known as cytoplasmic abundant heat-soluble and secretory abundant heat-soluble proteins. These act like molecular bodyguards, stabilizing other proteins, membranes, and DNA during dehydration or extreme cold. They help the tardigrades survive drying out by creating a glass-like state inside their cells, a process called vitrification. Interestingly, unlike many other organisms that relying on sugars like tree halos to vitrify, tardigrades do it with these special proteins, which form a protective glassy matrix. Their secretory proteins, especially the SAS proteins, also play a role in protecting cellular structures. They prevent membranes from fusing or breaking apart during dehydration, keeping cells intact. Experiments with model membranes called liposomes show that these proteins can shield membranes from damage caused by drying or other stresses. All these molecular tricks make tardigrades true masters of survival. Their DNA is protected by Dsup, repaired with TDR1, and their cells are stabilized by shapeshifting IDPs that form a glassy matrix. They don't rely on common sugars like tree halos but instead use these unique proteins to vitrify their cells and prevent damage. These tiny creatures have evolved a quirky set of molecular strategies that allow them to endure conditions that would wipe out most forms of life. Their secrets are not just fascinating but also open up exciting possibilities for future research in medicine, space travel, and even protecting human cells from radiation.